It's my privilege to get to introduce our keynote speaker tonight. Tonight, it feels like it's been a long day already. Bruce McEwen PhD, who is the Alfred E. Mirsky Professor and Head of the Harold and Margaret Millikan Hatch Laboratory of Neuroendocrinology at the Rockefeller University. I'm going to let him talk about his research, and I refer you to page 11 and 12 in your book for more about him and his abstract. But I want to give you a sense of what Dr. Scotto said about his contributions to science within his research, which he'll tell us about, but beyond. For one thing, he has wide recognition from his peers as a member of many prestigious institutes, such as the U.S. National Academy of Science and the Institute of Medicine, as well as a fellow of the AAAS and the New York Academy of Sciences. And for all you students in the audience, he has served as the Dean of Graduate School, as well as he is on the Scientific Advisory Board for Anti-Aging Games, which they have a great website. And if you have a chance to talk to him about how they're really utilizing the modern aspects of technology to engage not only the young people like my son that are totally addicted to video games, but those of us that are getting these iPads in terms of really thinking about these technologies and anti-aging and a broader impact on society. It's a really fantastic story. He exemplifies service to science and societies, having served as president of the Society for Neuroscience. Those are our great advocates with the government and with the community in terms of the importance of science to the life of everyone in this country. And he really shows how scientists can impact public health in his service on the MacArthur Foundation Research Network on Socioeconomic Status and Health, and as a member of the National Council on the Developing Child, focusing on healthy brain development as a key to physical and mental health. All of this service is really important, to making sure that the work that we're all dedicated to doing impacts on the lives of people in this country and around the world. And particularly to me, it's very important that in an age where we need to engage the public and politicians in the importance of science and education in the health sciences, he has written several books for the general public. He's really been a trendsetter in communicating science to the public, working with science writers such as Elizabeth Lashey on The End of Stress as We Know It. I personally subscribe to Reducing Stress. It would be very nice, as well as The Hostage Brain with Harold M. Shemick Jr. Lastly, I would like to say he was most gracious in accepting this invitation to speak to us, and everybody that's met him in the past remembers him and recognizes him as truly a gentleman scientist, an excellent advocate for all of us, and we are really looking forward to you engaging us with your work on the brain on stress. Dr. McEwen, thank you.
Thank you very much. Now I have to live up to all the nice words that are said about me. I might point out that I actually calculate that I have been a New Jersey resident for about 40 years, going in and out of the city. So I'm definitely with you here. So the topic is stress, and this slide sort of tells the story. Robert Sapolsky's wonderful book, Why Zebras Don't Get Ulcers, is contrasted with this New Yorker cartoon. These are the Fortnoys. The Fortnoys are the young worrieds. And apparently human beings, at least as far as we can tell, have this ability to worry about things, which can be good if you're planning ahead, something that we don't think a zebra necessarily has, although we can't get inside its head. But the idea that stress and anxiety, especially in the present day, are part and parcel of our lives. And how we deal with them, and in particular how the brain deals with them, is the topic of this talk. So when we think of stress, here's the adrenal glands that put out adrenaline and cortisol. Here's ACTH that comes from the pituitary to stimulate the cortisol secretion. Here are two neurohormones, CRH and vasopressin, corticotropin releasing factor or hormone, which is secreted from neurons in the hypothalamus in response to incoming neural input that can be from physical stressors or psychological stressors. And this cascade then initiates initially adrenaline secretion, part of the fight or flight response, and cortisol secretion. I think we all know about that. But what I want to emphasize is that acutely, as you might imagine, Mother Nature put it there to help us adapt and survive. And I'll say a little bit more about the first point, that it enhances immune function. It also improves particular aspects of memory, things that are involved in perhaps avoiding danger in the future if we survive, if the zebra survives, etc. Promote energy replenishment and more efficient cardiovascular function. These are its primary functions. Now, we're also aware of the fact that chronically too much glucocorticoids, well, we can use it to suppress an inflammation. It will also suppress overall adaptive acquired immune function. It can inhibit aspects of memory, and it can promote bone mineral loss, as in Cushing's disease, muscle wasting, and it's part of the metabolic syndrome. So we talk about inverted U-shaped dose response curves, and you'll hear a lot more about that. Now, because people tend to think about only the negative aspects of stress, and in particular on immunity, I wanted to emphasize something that comes from Firdaus Dabar, who's now a faculty member at Stanford in his PhD work, and then since PhD with us at Rockefeller. He showed that if you immunize a mouse or a rat with a chemical antigen, and then a number of weeks later, when an acquired immune response has occurred, you apply the antigen to one of the ears swells. That's because actually immune cells are trafficking from the blood into the ear because they are there to try to defend against a pathogen, if it were, or repair a wound. And what he showed is that this DTH, delayed type hypersensitivity response, lasts for a number of days. We know that when we have a tuberculin skin test or something like that. But if you acutely stress the animal at the time of applying the antigen, either with a moderate stress or with a severe stress, it enhances this response. Now, this is good if you're trying to defend against a pathogen, establish an immunization. It may be bad if you have an autoimmune disease. It could make matters worse, and we know that stress can make matters worse. Now, where the inverted U comes in is if you chronically stress the animal for a number of weeks. We use a restraint stress during the animal's resting period. It's not a painful stress. The animal is confined. It can interact with its cage mates during its dark, active phase. Nevertheless, it causes a change in the body so that then when you do this immune challenge, and there's a different y-axis here, you see a marked impairment of the DTH response. So here we see the downside of the effects of stress on immunity that we're probably all too familiar with thinking about. But I just want you, because this is taken, it's still something that people don't incorporate into their thinking. That's why I'm emphasizing it now. So keep that in mind as a prime example of this inverted U-shaped dose response curve. Now, glucocorticoids, of course, are primary activators of many things in the body, both positive and negative. And part of their effects involve direct effects on transcription of genes through a glucocorticoid response element, or one that's referred to as an NGRE, where it seems to inhibit transcription. But then there are indirect effects of glucocorticoids that are mediated by the tethering of the receptor to other transcription factors, so involving an interaction with a number of different signaling pathways. And there's another aspect which I won't have time to go into in detail, but has been occupying us as of late. Besides the direct and indirect genomic effects, glucocorticoid receptors are translocated into mitochondria, where at physiologic doses they actually help to buffer calcium and have antioxidant effects, neuroprotective effects, but at higher doses they do the opposite. They promote, this mechanism fails and the cell is endangered, as Robert Sapolsky term that he's used. Glucocorticoids directly work on membrane-associated mineralocorticoid and also, not shown here, glucocorticoid receptors to directly stimulate the release of neurotransmitters like glutamate. That's the major neurotransmitter in the brain. So this is a stimulation, and these receptors are the same receptors as work in the nucleus, except they've had some modifications and they sit near the membrane, and they do this in ways that are still somewhat mysterious. Glucocorticoids also stimulate through another receptor, still somewhat of a mystery, the synthesis of endocannabinoids. The brain's reason, the reason that marijuana is effective is that the body makes its own endocannabinoids, and they act presynaptically, and they inhibit the release of neurotransmitters. GABAergic in some cases, glutamatergic in other cases, and so forth. So they have an inhibitory effect on release. So what we're looking at is a whole spectrum of what we call non-genomic and genomic, including the indirect genomic effects that have broadened our understanding of how these adrenal steroids work. Now, adrenal steroids are not alone. They are part of a network which could be expanded beyond this to include metabolic hormones. The idea is that sympathetic, parasympathetic, glucocorticoid, DHEA is another adrenal steroid, inflammatory cytokines, anti-inflammatory cytokines, a certain number of free radicals which are essential for cell function. All of these systems these mediators talk to each other, influence each other, either directly or indirectly. So we're talking about a nonlinear network that is activated when something happens to the organism that we happen to call stress. And the other important point here is that all organ systems in the body, the brain, the immune system, the cardiovascular, renal systems, metabolic system, are all being simultaneously affected by this network of allostasis. Allostasis is the active process that the body uses to maintain homeostasis, which of course is stability and for survival. Now, the brain is central to all of this. And I'm going to talk about different parts of this model, which was from a 1998 paper at New England Journal that talked about this model of allostasis leading to adaptation and allostatic load, which is the negative but almost inevitable consequence, the wear and tear on the body and the brain of being exposed to repeated stressors. So we're talking about accumulation of load or weathering with aging, but also something we'll come to which we can refer to as biological embedding that relates to what happens in those early experiences that are modifying the genetic constitution that we have and now what we refer to as the epigenome. So we'll come back to that. Let's talk about stressors. They come from things like trauma and abuse. We'll talk about that, major life events, and also the daily stressors of our working and home and community life. How do we talk about stressors? Well, the way I like to define them, we talk about good stress or positive stress, where we may feel some exhilaration from a challenge that has a satisfying outcome, like in my case, hopefully giving a decent talk today. A sense of mastery and control. You have to have good self-esteem, good sense of that you can do it. Tolerable stress is something that happens to people who have good self-esteem, but a bad thing, a major life event. And yet, if you have those good internal mechanisms and you have good social and emotional and material support, you can weather the storm and recover. Resilience, recovery. And then there's something called toxic stress, which almost speaks for itself. Often there is a lack of adequate self-esteem, perhaps unhealthy brain architecture, maybe as a result of both genetic risk or early life adversity. We'll talk about that. And it's and as a result in early life adversity, chaos in the home, abuse and neglect can contribute to the failure to develop the good neural architecture and good self-esteem. And we'll come back to that. Now, many of you perhaps have heard about the Adverse Childhood Experiences Study by Vincent Felitti and Robert Anda. The CDC was responsible for the study, but it was done in the Kaiser Permanente health system in California in a middle-class population. This is not poor people. These are middle-class people, and yet as a function of chaos in the home, harsh language without adequate positive reward and support, instability of emotional control, perhaps related to parents' own instability, uncertainty in their personal lives or professional lives, physical abuse, sexual abuse. These represent a scoring system, the ACE score, and the higher the ACE score, the greater the incidence of things like heart disease, increased frequency of smoking, obesity, and diabetes, drug abuse, higher risk for AIDS, depression, anxiety, anger control, and other kinds of antisocial behavior. And this is happening all over the world in middle-class populations, in upper-class populations, related to the nurturing environment or lack thereof early in life. So we'll come back to that. Something we're all aware of, this was these were the famous Avshalom Caspi studies-based studies in New Zealand, remarkable set of studies that showed that alleles of common genes like monoamine oxidase or the serotonin transporter influence whether, in this case, childhood abuse will be transmitted from abuser to the child. It's often said that an abused child will seek a partner who resembles the abusing parent. How often that happens is influenced by the genotype as well as by the experience of being abused. Serotonin transporter alleles influence vulnerability to life stress and causing depression. I think many people are aware of these kind of things. Well, how do we think about these genes? There's a good side and a bad side. A wonderful Atlantic article by David Dobbs talks about orchid children, the ones who are sensitive. I'll say more in a minute, as opposed to dandelion children, the ones that are tougher. And Bruce Ellis and Tom Boyce have done academic, very careful work and published many papers on this. And one of the papers from one of the slides I got from Bruce Ellis not too long ago talks about high reactivity. These are the orchid children, or high reactivity, the solid line, the orchid children, and low reactivity, more the dandelion children. In terms of externalizing behavior, in a low conflict situation, the high reactivity kids do better, have less externalizing, but in a high conflict situation, they're a lot worse. So they are adversely affected like an orchid would be grown in a bad environment. In terms of more positive things, if we're talking about fall school engagement, the high adversity, the high reactive children, the reactive children with the reactive genotype do very well in a low adversity environment, but do much more poorly in a high adversity environment. So this illustrates what is meant by these reactive alleles, which of course we're only beginning to recognize the many different possibilities in the human genome. So let's talk a little bit about allostasis, the idea that it's the normal process putting out cortisol, adrenaline, inflammatory products that lead to adaptation, and the inevitable consequence of having a lot of stress or dysregulating, not putting out enough cortisol when you need it, having too much inflammation, having an imbalance in this network I talked about in that very colorful slide, leads to wear and tear or weathering and increased incidence of disease, as in toxic stress. But there's a behavioral component too, which relates to when we feel stressed out. What happens to us when we feel overwhelmed, out of control, exhausted, anxious, frustrated, angry? We may lose sleep at night. We eat too many of the wrong things, drink too much alcohol, smoke if we're so inclined, or neglect regular moderate exercise because we're sitting in front of our computers trying to finish a rant, answer on Facebook inevitably. I mean, there are many ways that we can be sedentary and do things, and yet then neglect moving around, even walking around the block if you, or doing something physical. All of these contribute to allostatic load. So psychosocial stress is a major contributing factor. And then we come to the brain. And this is where a lot of our work over a number of decades has taken place. And I'll tell you just a little bit about this. I'll talk briefly about 3 brain areas: the hippocampus, which is an organ for the memory of daily events, spatial memory, and mood regulation. It also helps shut off the stress response. The amygdala is a brain region of anxiety, fear, and aggression that actually helps turn on both the autonomic and the HPA stress response. And then the prefrontal cortex, which has a top-down influence to control both the autonomic and shut it off HPA stress response, but is also important for decision-making, working memory, self-regulatory behaviors, mood, and impulse control. And its development and function is critical, especially its deficiencies in development and function is critical in the case of the toxic stress that we'll talk about. Now, we discovered that the hippocampus, shown here, Amon's horn and dentate gyrus, has receptors for adrenal steroids. They have the genomic receptors. They also have these non-genomic actions that I was talking about earlier. We know now that the hippocampus is important not only for memory functions, but also for mood regulation, and it's a target of depression and antidepressant treatments. We found, well, others have found that the CA3 region, there's this trisynaptic circuitry. The input from the cortex comes in to both the dentate gyrus and the CA3. This is the input that degenerates in the early stages of Alzheimer's disease. This input is involved in a feed-forward feedback circuit that's very sensitive. It's part of what enables the hippocampus to remember things in space and time. If you overstimulate it, it's vulnerable to develop seizures, which causes permanent irreversible damage or loss of neurons in this CA3 region. And the CA3 normally projects to CA1 and then out of the hippocampus to other parts of the brain. Now, we found that repeated stress causes the apical dendrites of these neurons to become shorter and less branched. The basal dendrites don't change. This is a reversible process if you stop the stress in time. So this is not brain damage. At the same time, or yes, in about the same period of time, and this is actually due in large part to Elizabeth Gould, who came as a postdoc in lab, introduced us to some of the anatomical methods, and she and Heather Cameron rediscovered neurogenesis in the dentate gyrus that had been somewhat neglected, although reported by other investigators decades before. This neurogenesis is ongoing through adult life, can be reduced by stress, activated by things like regular voluntary exercise. So there's this plasticity in these parts of the brain, and we were particularly interested in this aspect and found others had reported that during a restraint stress, the same kind of thing that we used for this delay type hypersensitivity, if you do it repeatedly, you not only suppress DTH, you also cause this dendritic remodeling. During a restraint stress, there's an increase in extracellular glutamate, but which does not occur in an adrenalectomized animal. So that's the basis of an interaction between circulating adrenal hormones and the release of glutamate. And I've said some things about mechanism. As a result, the effects of repeated stress can be prevented by blocking glucocorticoid synthesis. They can also be blocked by blocking NMDA receptors, by using lithium and dilantin, all of which affect excitatory amino acid transmission, by certain antidepressants, and by benzodiazepines, which enhance the inhibitory part of the transmission, and also by a deficiency or overexpression of BDNF. If you have too little, you can't shrink your dendrites, they're already shrunken. If you have too much, you can't shrink your dendrites because they're already expanded. So it's like a floor and a ceiling effect. So excitatory amino acids work with glucocorticoids and a host of other mediators. In terms of the neurogenesis, a PhD thesis by Cara Pham in about 7 or 8 years ago showed that there is, in fact, repeated stress, but we had to go out to 6 weeks of this daily restraint, caused a decline in neurogenesis here. And if you extended it out to 6 weeks, you could find a significant reduction in the number of granule cells and the volume of the dentate gyrus. In other words, chronic stress could shrink the hippocampus, of that part of the hippocampus. So that's the other part of this. As I said, we now know that there are both genomic and non-genomic actions. I mentioned them earlier. I won't spend any more time on this, but if anyone wants to ask me about this, I'll be glad to elaborate. But the bottom line, if we're thinking about, again, our inverted U, acute activation of this system is important for increasing excitability, improving certain aspects of memory, and interaction between excitatory amino acids and adrenal steroids. A higher level acutely of stress and stress hormones can inhibit excitability, lead to long-term depression, impair certain aspects of memory. These are all acute effects. Repeated stress leads to what we call adaptive plasticity, the suppression of neurogenesis, which is reversible, and the remodeling of dendrites that I spoke about, which is reversible at least in a younger animal. I'll say something about aging. And then, as Robert Sapolsky and others have shown, if you have a stroke, a seizure, or head trauma, the glucocorticoids and excitatory amino acids actually produce the permanent irreversible damage and cell loss. Now, other people have taken these findings about the sensitivity of the hippocampus and shown that it becomes smaller in major depression, smaller in type 2 diabetes. And Antonio Convite at NYU has shown repeatedly that children with type 2 diabetes have major problems in their brain circuitry. Their brains are really adversely affected. Adults also the same. Type 2 diabetes is also a risk factor for later dementia. So that's a very severe problem, and diabetes has to be recognized as, in part, a brain disorder. Post-traumatic stress disorder is associated with a smaller hippocampus, perhaps as a predisposition, maybe as a reflection of low self-esteem, which has been shown to lead to a smaller hippocampus. But the experience of trauma can lead to a further shrinking of the hippocampus and also incidence of depression, as well as this special anxiety disorder. Cushing's disease is associated with a smaller hippocampus, with mood impairment, with memory impairment. The good news there is that there is at least some partial recovery from correcting the hypercortisolemia, so there may be some plasticity there. But you don't have to look at disease. And a 20-year history of chronic stress, chronic perceived stress, is associated with a smaller hippocampus in a cross-sectional study. Chronic jet lag, not having enough time to recover between international flights 5 days or less in a study of, I think, Singapore Airlines, was associated with a smaller hippocampus and impaired memory, whereas if the crew had 14 days or longer, they didn't show these effects. Lack of exercise, I'll say about in a minute, is associated with a smaller hippocampus, and chronic inflammation, which is a sort of common denominator of most of the diseases of modern life, including diabetes, depression, and so on, is associated with a smaller hippocampus. Now, the positive news is that regular moderate exercise can increase hippocampal volume even in older people, especially in older people. Intense learning will actually increase the volume of the hippocampus. And some studies, there need to be more, showing that antidepressant treatment chemically may be able to do that. Exercise is one of the best antidepressants, I should add. This study by Kirk Erickson and Art Kramer's group at the University of Illinois showed that if you get sedentary adults in their 60s and 70s to walk an hour a day, 5 out of 7 days a week, over a year, it enlarged both the left and the right hippocampus. That was the area of the brain most affected. And so physical activity is one of the best remedies, not only for cardiovascular disease, for also brain function and anti-aging treatment. And you might be amused by this slide that somebody gave me. It says, my grandmother started walking 5 miles a day when she was 60. Now she's 97 years old, and we don't know where the hell she is. So let's talk about several other areas of the brain. Just as the hippocampus neurons become shorter and less branched in a reversible way, the same thing happens with the medial prefrontal cortex. But in the amygdala, the same stress will cause neurons in the amygdala to become larger. And the same thing happens in the orbital frontal cortex. So the brain circuitry becomes imbalanced. More anxiety, more vigilance, less ability for top-down control, less ability for memory. Let's talk about the amygdala. In a recent study that we've collaborated with Sumantra Shona Chatterjee, who's in Bangalore, India at the National Center for Biological Sciences, he's shown that chronic stress a single episode of stress, not just repeated stress, will lead 10 days later to increased levels of anxiety, shown here in a plus maze, and increased density of spine synapses on basolateral amygdala neurons. So there's a delayed increase in anxiety, a delayed increase in synaptic connections. The dendrites don't get longer. You need to do repeated stress to have that happen. But this seems to be sufficient to increase anxiety. And what we did in a collaboration that's just being published in Biological Psychiatry is to show what we did was we basically said, let's manipulate this with drugs. So we gave a saline injection, which you have to do if you're going to give a drug, you have to have a control. Damned if the saline injection prevented this whole thing, if it was timed just before the animal went into this immobilization apparatus. And we showed that basically this was because of a timed elevation of glucocorticoids, a very modest elevation, but it had to be timed right. Now, where this may be of relevance are studies first in epidemiology showing that people with low normal levels of cortisol are more vulnerable to PTSD. Other studies in a clinical research setting by Gustav Schelling in Germany that people undergoing open heart surgery often develop PTSD-like symptoms. This can be reduced by increasing their adrenal steroid level during surgery to the maximum sort of physiologic output of the adrenal. Another animal model by Hagit Cohen and colleagues in Israel, and then this paper that talked about that animal model, but also talked about a study where car accident victims in Israel were taken into the clinic and within several hours given a bolus of a glucocorticoid. They showed less incidence of PTSD. So it may be that like quelling an inflammation or containing an inflammation, and it's probably not because of inflammation in the brain, that glucocorticoids are important as protective agents. Now, there are studies by Pete Generos in Pittsburgh showing amygdala activity to faces. If you see an angry or sad face, your amygdala gets turned on, your blood pressure and heart rate may increase, your HPA axis may increase, and people with early stages of atherosclerosis tended to show higher levels of activation of the amygdala with those angry and sad faces, suggesting a very gradual process of exposure because of a more sensitive system that may actually lead to a form of allostatic load in terms of early stages of cardiovascular disease. Studies of the prefrontal cortex, a wonderful collaboration with John Morrison and Patrick Hoff in New York. We found that this shrinkage of the medial prefrontal cortex is reversible in young adults, but is lost in animals as they age. They still show shrinkage of dendrites, but they don't recover as fast, if at all, when you stop the stress. And I'll talk about two more things in the next few slides. The next slide is one that extends the work on the rodent model to medical students studying for a board exam. Connor Liston, who did the animal studies, teamed with B.J. Casey, looked at volunteer medical students, defined a circuit using fMRI that was activated by a task that was similar to one that Connor had used in rats to show impaired cognitive flexibility in the rat brain, found that this circuit involves the prefrontal cortex, used the perceived stress scale, which essentially gets at how stressed out you feel in terms of personal things, all things that are going on in a person's life, including the academic aspects in the case of the medical students, found that higher scores on the perceived stress scale predicted poor mental flexibility, and they also predicted poor functional connectivity in the circuit. Now, they couldn't look at synaptic connections, dendrites, spines, and so on, but this is what you'd predict. Now, the good news is that these effects wore off after a vacation. Took the exam, they had a month off. So at least the young brain is resilient. Well, about older people, but individuals do differ enormously in the degree of plasticity that their brains and bodies will show. Another study by Ilya Karatsarios, who's an expert in circadian, postdoc expert in circadian biology, trained with Ray Silver, showed that if you put a mouse into a 10-10 light-dark cycle, that their biological clock in the suprachiasmatic nucleus cannot adjust to. And they're sort of free running. Their temperature rhythm is free running. It's like a shift work model where you're continually shifting your sleep and waking pattern. Led in a month or so to increased body weight, increased insulin and leptin, suggesting insulin and leptin resistance. And the medial prefrontal cortex neurons showed the same debranching that we saw after chronic stress, and the animals showed a cognitive rigidity or inflexibility. So circadian variation, sleep deprivation, on top of that, I mentioned jet lag, are all factors that do affect brain circuitry. And finally, females differ from males in very subtle ways. And one of the ways shown by Rebecca Shansky, working with John Morrison and us, is that females do not show the dendritic remodeling seen in males. At least the neurons that turn out to be projecting cortically from the prefrontal cortex do not change in females as they do in the male. But there's a set of neurons that project to the amygdala. And females, but not males, showed an expansion under stress of those dendrites, of those neurons that project to the amygdala. And that was dependent on having estrogens on board during the stress. So we don't know, no one knows the consequences of this, but it suggests that there may be many other subtle sex differences that distinguish the female from the male brain, and yet we know that males and females do many of the same things, pretty much equally well. There are some biases in one direction or the other, but they may do so through different strategies and even different underlying mechanisms. And this is a huge area that needs to be expanded in the future. Art Kramer showed that not only does the hippocampus improve with regular moderate exercise, but blood flow and decision-making capacity is improved by the same pattern of regular moderate exercise. And finally, another example from the human domain is if you will encourage people to volunteer, as was done in the Experience Corps in Baltimore by Linda Freed at Hopkins and colleagues, to volunteer as elderly people as teachers' assistants in the public elementary schools. A little bit of car fare money. They formed a group. They were trained. They performed work as, help with reading and discipline and library and so on in the classrooms. And that improved the educational process. But another study that involved also Art Kramer's lab showed that their prefrontal cortical blood flow, their executive function improved, as well as their mood, and their overall physical health did not decline as much as a control non-volunteer group. So this is where one combines physical activity with some kind of meaning and purpose in life, and is the basis of why I'm interested in this anti-aging games activity to encourage older people. Okay, let's get back then to, for the rest of the talk, to the early childhood intervention. What is toxic stress? Well, chaos in the home, a sense of helplessness and distress leads to poor self-regulatory behavior, which may mean poor prefrontal cortical development. We know it develops during this period after birth up through adolescence into young adulthood, faster in females than in males, which is why males have such high car insurance rates. Obesity is also a result, along with cardiovascular reactivity, of early life adversity, and systemic inflammation is an almost universal consequence of ACE, of adverse childhood experiences. Risky families also lead to many of the same consequences. These are cold and unsupportive families, part of the adverse early experiences. Lack of verbal stimulation leads to poor language skills and poor executive function. So early verbal stimulation, the serve and return between the parent or caregiver and the child, that leads to verbal ability and emotional control. And of course, I've already talked about abuse and neglect. Now, I want to show you a brief video from the National Scientific Council. Let's see.
Learning to deal with stress is an important part of healthy development. When experiencing stress, the stress response system is activated. The body and brain go on alert. There's an adrenaline rush, increased heart rate, and an increase in stress hormone levels. When the stress is relieved after a short time, or a young child receives support from caring adults, the stress response winds down and the body quickly returns to normal. In severe situations, such as ongoing abuse and neglect, where there is no caring adult to act as a buffer against the stress response stays activated. Even when there is no apparent physical harm, the extended absence of response from adults can activate the stress response system. Constant activation of the stress response overloads developing systems, with serious, lifelong consequences for the child. This is known as toxic stress. Over time, this results in a stress response system set permanently on high alert. In the areas of the brain dedicated to learning and reasoning, the neural connections that comprise brain architecture are weaker and fewer in number. Science shows that the prolonged activation of stress hormones in early childhood can actually reduce neural connections in these important areas of the brain at just the time when they should be growing new ones. Toxic stress can be avoided if we ensure that the environments in which children grow and develop are nurturing, stable, and engaging.
So back then to this, which we've seen before, and let's then there are animal models, I won't go into details about them, that show something about the neural basis of attachment in spite of abuse, involving the amygdala, deleterious effects of maternal anxiety in terms of food insecurity on the part of the mother transmitted to the child, leading to increased anxiety and also possibly metabolic syndrome. The intrauterine environment, very important. The important study started by Victor Denenberg, Gig Levine, carried on elegantly by Michael Meany's lab and Moshe Ziff, have emphasized the importance of maternal care in rodent models, David Lyons in primates, and have emphasized, and work by Acacia Tang, a younger colleague at the University of New Mexico, has emphasized in those rodent models the importance not only of the amount of maternal care, but of the consistency over time and the benefits of novel experience. And if any of you are interested, I refer you to a number of relevant papers on very interesting animal models. But we'll talk a little bit about the epigenome, the idea that the unwinding of DNA makes it easier to read. There are factors, histones, and the modification of cytosine residues by methylation that affect the readability of this. And in fact, the idea that putting on a methyl group may increase repression of a particular gene, in some cases the opposite. There's a whole complex code that is being gradually uncovered. We talk not only then about the genome, but the epigenome, and the notion that identical twins are genetically identical, but actually even early in life can show some differences, but these differences diverge as they get older. And this paper in PNAS talking about the methylation patterns on chromosomes, showing that a pair of young identical twins have very similar patterns, but when the twins are in their 50s or 60s, it's largely divergent. So this is part of the reason why there is not identical outcomes in monozygotic twins in terms of disease and other issues that develop across their lifetime. And this, of course, is a huge, important area for current and future exploration. So what can one do about it? Well, the National Scientific Council on the Developing Child, and I recommend their website, based at Harvard, but involving scientists from all over the country, emphasizes that adult disease prevention, as in the ACE study, begins with reducing early toxic stress, that early childhood programs benefit lifelong health, not just education. Think about the diabetes connection, which we realize is a huge, huge problem for the brain as well as the rest of the body. Promoting physical health benefits the brain. And parent-child assistance, as in the nurse-family partnership, is certainly one way of helping to develop early life. The Harlem Children's Zone Baby College is another attempt at a group level. There is desperate need for using these programs and for developing others. Now, I've already mentioned a number of top-down therapies, not talking about the fact that pharmaceutical agents are important, but top-down therapies that involve integrative activity of the nervous system, such as regular physical activity, social support and integration, cognitive behavioral therapy that can actually change the brain, just as exercise appears to change the brain. And one example of this is this study from the Harvard Mind-Body Center that people with a chronic anxiety disorder who respond to a mindfulness-based cognitive behavioral therapy with reduced emotion actually show a reduction in the volume of the amygdala. And there are other studies that are suggesting that these behavioral manipulations, putting drugs aside, actually can benefit brain and structure, function, and behavior. And then there's a new area that was started in studying the so-called lazy eye phenomenon, monocular deprivation, which was thought to be an irreversible condition, but giving, of all things, fluoxetine to adult animals that had only monocular vision, while stimulating the visual system actually reversed that and created more of a binocular pattern over a relatively short period of a number of weeks to a month. This was then replicated by the same group using an intermittent food restriction paradigm, which, among other things, elevates glucocorticoid levels. Every other day, the animals were not fed. And that, in turn, could be replicated by giving glucocorticoids in the drinking water every other day while visually stimulating. So this, what we thought was an irreversible change due to development, was subject to activation by agents which we think are actually enhancing the production of growth factors like BDNF and FGF and so on that facilitate plasticity. A Lancet study showing that people recovering from stroke and doing their physical therapy, actually, if they take fluoxetine, they will show a significant recovery. And this is also the strategy that is used by psychiatrists in the treatment of depression, whether it's an antidepressant drug or physical activity. The combination of that with a targeted behavioral intervention seems to be essential for pushing the plasticity in the right direction. So I guess what I want to say in sort of summary of this new view of plasticity is that the adult brain does show plasticity, and I think we're really only beginning to recognize its potential. Now, whether this potential can be directed in the case of people who have early life adversity and these rather long-lasting conditions, of course, I think the biggest challenge of all. So what I've tried to summarize for you is the notion that there's protection and damage by the mediators of stress and adaptation, that the brain is a target of changes in brain architecture that also alter physiology and behavior, that there are powerful effects of the social as well as physical environment and the person's own personal history that we have to factor in. Biological embedding, as in adversity, affects the course of this cumulative change, that is the degree to which weathering or allostatic load will take place, the importance of what I call top-down interventions, and the potential, still somewhat untapped, of brain plasticity. And finally, as in a symposium like this, breaking down silos of knowledge within medicine and across disciplines is extremely important. People have to talk to each other, collaborate with each other. My experience on MacArthur Foundation networks and shows that you can get people to attack a problem and bring all sorts of skills to provide some novel approaches and possible solutions. A large group of people currently in the lab, collaborators in other institutions, former students and postdocs and colleagues, and then the MacArthur Foundation, the National Scientific Council, and of course been supported over the years by NIH grants. Thank you very much.
At this point, we're going to take first a minute to thank our speaker for putting such a wonderful context to the rest of our day here at the symposium. Thank you, Dr. McEwen, for joining us today.